In this episode, we'll explore genetic inheritance. Key topics we'll discuss today include how genes control characteristics, different forms of genes called alleles, genetic diagrams, and family trees. Many genes can directly control what characteristics you develop. Different genes control different characteristics. Some characteristics are controlled by a single gene, like mouse fur color and red-green color blindness in humans. However, most characteristics are controlled by several genes interacting together. This makes inheritance patterns more complex for traits like height and skin color. All genes exist in different versions called alleles. These are represented by letters in genetic diagrams, one letter for each allele. You have two versions or alleles of every gene in your body, one from each parent. These alleles can be the same or different from each other. If an organism has two alleles for a particular gene that are the same, then it's homozygous for that trait. If the alleles for a particular gene are different, then it's heterozygous. Sometimes one allele can completely override the other when they're different. The allele that shows itself is called the dominant allele. We typically use a capital letter for dominant alleles. For example, capital A. The other one is called the recessive allele, represented by a small letter, for example, lowercase a. A recessive allele only shows itself when there's no dominant allele present. For an organism to display a recessive characteristic, both its alleles must be recessive. To display a dominant characteristic, the organism only needs one dominant allele. This means the dominant allele overrides the recessive one if the organism is heterozygous. Your genotype is the combination of alleles you have, while your phenotype are the observable characteristics you have. Let's see how we can predict inheritance patterns using genetic diagrams, also known as Punnett squares. Suppose we're breeding chickens with long legs. The allele which causes chickens to have long legs is recessive and represented by a lowercase h, while normal legs are due to a dominant allele represented here with a capital H. Let's look at what happens if you breed from two heterozygous chickens. We start by identifying the parents' genotypes. Both parents are heterozygous. Next, we determine what types of sex cells each parent can produce. We can then create a Punnett square to show all possible combinations. We place one parent's potential gametes along the top and the other parent's along the side. Filling in the square gives us the following. Looking at the phenotypes, we can see that 3 of the offspring will have normal legs, while 1 will have long legs. This gives us a 3-to-1 ratio of normal to long-legged offspring in this generation. Now, if 2 of these offspring breed together, what happens in the next generation? It depends on which offspring breed together. If a homozygous dominant breeds with a heterozygous chicken, all their offspring will be normal. On the other hand, if a heterozygous chicken breeds with a homozygous recessive chicken, half their offspring will be normal and half will have long legs. Let's look at another example. A lion with long hair, which is the dominant trait, is bred with a lion with short hair, which relates to the recessive trait. The long-haired lion has a heterozygous genotype, while the short-haired lion has a homozygous recessive genotype. When we work out the Punnett square, we find the offspring will have a 1-to-1 ratio, half with long hair and half with short hair. This happens because the heterozygous parent can pass either the dominant or recessive allele to its offspring. The homozygous recessive parent can only pass the recessive allele. Understanding genetic inheritance can help us interpret family trees, which is particularly useful for tracking genetic disorders like cystic fibrosis. From a family tree, we can often tell if an allele for a particular disorder is dominant or recessive. For cystic fibrosis, we know it's a recessive disorder because plenty of people carry the allele but don't have the disease. By analyzing a family tree, we can also determine the probability that someone will inherit a genetic disorder. For example, if both parents are carriers for a recessive disorder but don't show symptoms, there's a 25% chance their child will have the disorder. Similarly, there's a 50% chance their child will be a carrier like them, and a 25% chance the child will neither have the disorder nor be a carrier. The risk calculation for the new baby is just the same as in the genetic diagram. So a new baby could be unaffected, be a carrier, or have cystic fibrosis. Genetic diagrams are important tools in understanding and predicting inheritance patterns. They allow us to calculate the probability of particular traits appearing in offspring based on the parents' genotypes. These diagrams help us understand why certain traits might skip generations or appear unexpectedly. For example, if two brown-eyed parents have a blue-eyed child, genetic diagrams explain that both parents must carry a recessive allele for blue eyes. It's important to remember that genetic diagrams show probabilities. They don't guarantee exactly what will happen with each individual offspring. Just like flipping a coin doesn't guarantee exactly 50% heads and 50% tails, in a small number of flips, genetic inheritance can sometimes produce unexpected ratios in small family sizes. In reality, many traits are influenced by multiple genes and environmental factors, making inheritance patterns more complex than the simple examples we've discussed. However, these principles form the foundation for understanding how genes are passed from one generation to the next. In our next episode, we'll explore how these inheritance patterns can lead to genetic disorders when certain mutations are passed down through families.